So we've covered DNA, and now in this unit, we're going to learn how DNA can be used by humans for particular purposes. So what is biotechnology? Biotechnology is the use of technology to manipulate biological organisms. It often involves the use of microorganisms like viruses and bacteria to benefit humans. Biotechnology is not new. The use of mold to produce the antibiotic penicillin is an example of biotechnology that was begun in the 1930s. But we've made a lot of advancements, and biotechnology is being used more frequently than it was back then. Many types of modern biotechnology, however, rely on the ability to analyze, cut, and manipulate strands of DNA, which is called DNA technology. So this scientist is using biotechnology to determine the structure of the COVID-19 virus. What is the Human Genome Project? DNA technology began when a group of scientists from 20 institutions worldwide began trying to identify the entire DNA sequence for humans, which is called our genome. The goal here, after we determined that DNA was the genetic information of life, and then we determined what the structure was for DNA and how it was used, scientists assumed that they would be able to figure out what every gene in the human genome coded for, and then from there we could work backwards. When proteins weren't working correctly, we would be able to identify what gene was causing a certain disease or a certain deficiency and then correct that. So identifying the sequence could help scientists find genes that caused certain traits and diseases. The project began in 1990 and ended in 2003 when they had successfully sequenced 90% of the whole human genome. So this was a global project with lots of scientists from different countries and institutions working together in order to accomplish this as quickly as possible. Unfortunately, the Human Genome Project raised more questions than it solved. Scientists have determined since that only about 2% of our genome actually codes for proteins. Although some of the remaining genes are non-coding, like promoters, transcription factors, etc., over half of the human genome is made of repeating sequences with no known purpose. So if we were to break this down into percentages, there's about half of the human genome, all of the DNA sequences in a typical human, are these repeating sequences that were surprising to scientists. And then there's a large section of non-coding sequences, regulatory sequences, introns, which we've discussed, are spliced out to create the finished RNA product. And then just 2% is that protein coding region. So this was not what they were expecting. They were expecting all 100% of the genes to be coding for particular proteins. So if we continue on with what we've found from that point on, we start discussing something called epigenetics. So in the early 2000s, studies began to show that gene regulation, rather than the genome itself, caused many of the inherited illnesses scientists were looking to cure. So the regulation of which genes were being turned on and turned off was actually causing more traits and diseases than the genome, the gene sequence itself. When methyl groups, methyl being CH3, are added to DNA, The chromatin condenses on its histones, and the DNA is not able to be transcribed. So these little groups of carbon and hydrogen are called methyl groups, and when they are attached to DNA, pieces of DNA, double helix, the chromatin, or those long strands of DNA, actually condenses on the histones, winds the chromatin up more tightly, and then the DNA is not able to be transcribed. And that gene regulation process is called epigenetics and can be passed from one generation to the next, causing inherited traits and diseases. Epi meaning above or on top. So this is above the typical set of genes. This would be a next layer of traits and characteristics. So here's what that might look like if this is your DNA double helix. If a methyl group was added to that double helix, the histones would wind up more tightly, and then that DNA was going to be inactive for this transcription. So it's not going to be able to be separated and used as a template for creating proteins. Sometimes there are epigenetic factors that then allow that DNA to unwind, and then the genes are activated and they're able to be transcribed by the RNA polymerase. Epigenetics is interesting because it can be changed more rapidly than the genetic code, allowing an organism to adjust to its environment more quickly. We discussed the genetic code being caused, the changes in the genetic code being caused by mutations, so changes to the DNA bases, and that can happen very infrequently, and it can take a long time, and often it's harmful rather than helpful. So epigenetics is a new layer of a way that an organism can adjust to its environment and ways that traits can be inherited by gene regulation rather than the actual genes itself. Mother rats that lick their young can change the epigenetics of their babies. The licking activates a gene that helps the rats to adjust to stress more easily. So this was an experiment that scientists did on epigenetics to determine how traits were being regulated. And mother rats that licked their mice babies actually changed that the methyl groups, allowing some of that DNA to be transcribed and then creating proteins that allowed that mouse to be calmer. And then mother mice that did not lick their rat babies did not have babies that had that gene available, and so the proteins were not produced, and the mice remained anxious rather than calm. So in summary, the Human Genome Project helped scientists determine that the DNA sequence of a genome is only part of the cause for traits and diseases. Epigenetics, which involves regulating genes through the addition of methyl groups, can also affect the expression of genes. So if biotechnology is the ability for scientists to manipulate genetics or DNA, one of the ways that they do that is by cutting pieces of DNA for a variety of reasons. Bacteria naturally contain molecules called restriction enzymes, which can cut the DNA of viruses trying to infect them. So this is a protection defense for the bacterial cells. If a virus is trying to infect them and it inserts DNA into the bacterial cell, the restriction enzymes of the bacteria can cut that virus DNA, and therefore the virus then is no longer able to reproduce. Scientists can also use these restriction enzymes to cut DNA at specific locations in a genome. So using this same system, they can cut DNA pieces of a variety of different organisms at locations that allow them to separate these pieces into different sizes, insert genes, and other processes that they use to edit a genome. So BAM-H1 is a restriction enzyme that cuts wherever it finds a sequence like the one on the left. So it cuts right here, and it creates a nick here where there's a tail on one side of the DNA strand and a tail on the other side of the other piece. And that allows genes to be inserted into that DNA strand. And restriction enzymes are named for the bacteria from which they originate. So this is a bacillus restriction enzyme. It gets the name BAM. So using those cut pieces of DNA, we can then analyze the DNA. In order to separate and analyze the fragments of cut DNA, a process called gel electrophoresis is used. And this technique uses an electric current to make charged DNA fragments travel through a thick gel. So here you can see the gel electrophoresis chamber here, and the gel is this thick, clear substance in the middle. And these are pipettes full of pieces of DNA that are then loaded into little wells in the gel. And then the electrodes are turned on, and those pieces of DNA will travel towards one end of the electrophoresis chamber. The DNA fragments separate by size. Small fragments travel the furthest, and large fragments get caught in the gel and travel the least. The pattern of bands created by the different DNA fragments is called a DNA fingerprint. Here's an example of a DNA fingerprint. And these bands are locations of genes that have traveled through the gel. Unfortunately, gel electrophoresis is a painstaking and slow process. So scientists have recently come up with a new process of analyzing DNA that's faster and simpler. In the 1990s, genetic researchers determined that the non-coding regions of DNA had short repeating sequences called short tandem repeats, or STRs. So remember, we discussed that not all of the portions of DNA are used to create genes. There are non-coding regions, and that's where they found these short sequences that are repeating over and over again, called STRs. And although 99.9% of the genome is the same from person to person, the STRs vary widely, making them a great tool for identifying an individual. And STR analysis is now used as a more efficient way to make a DNA fingerprint. So this is an example of a DNA fingerprint created through STR analysis. This is similar to the DNA fingerprint on the previous slide, made through gel electrophoresis. It's identifying an individual, but this is made obviously by a computer, and it is analyzing those STRs to identify or to compare or analyze the DNA of different individuals. So this might be one individual, this would be another individual, and these peaks are representing those STRs. So they would match up if the two individual sequences were the same. How can we artificially copy DNA? So sometimes scientists have too little DNA for an experiment, and they can make copies of it with a technique called polymerase chain reaction, or PCR. So let's say that you are a scientist, a forensic scientist that collected DNA left behind at a crime scene. There might not be enough DNA left at that crime scene in order to do all of the different tests required to identify an individual. And so making a copy of those DNA sequences can actually help scientists have enough DNA to do all of the processes and experiments that they would be required to do. And this machine is known as a thermocycling PCR machine. So this is where the DNA would be inserted and copied. So let's go through the steps of the PCR process. It involves repeated cycles of heating and cooling to break and reform the DNA strands. So here is the piece of parent DNA that might have been collected from the crime scene. And the first step is to denature it. So it is heated, the DNA is heated, and it actually causes those two strands to separate. They unwind and they separate from each other. The second step is annealing, where primers are added to the solution of DNA and the DNA is cooled back down. And then in step 3, the DNA sequences are extended using something called Taq polymerase, an enzyme called Taq polymerase, and it adds more bases to finish the daughter strands. Taq polymerase is similar to DNA polymerase, which we discussed in DNA replication, but it works better in hot conditions, so we're going to raise the temperature of that solution again so that this DNA can be extended, and then eventually we'll end up with 2 copies of that original DNA strand. The cycle is repeated until enough DNA is formed to run the necessary tests. So in summary, DNA can be cut with restriction enzymes and analyzed through gel electrophoresis or STR analysis to make a DNA fingerprint. PCR can amplify the amount of DNA if only a small sample is available. So now that we've learned how to analyze DNA, how is this technology used? Well, DNA analysis has many practical applications. First, it can be used to identify relationships. So by matching the DNA profile of an individual with other family members, ancestry can be identified. Applications of this include paternity testing, establishing relationships in a family tree, like in 23andMe or Ancestry.com, and showing the evolution of a particular species. So how are different animals related to each other based on how their DNA matches up. In the paternity test to the right, the mother contributes half of the genes to the child. And so here's the mother. This is her DNA fingerprint. This is the child's DNA fingerprint. And then there are 3 potential fathers that are being tested to determine which contributed to the child's other genes. So this would be what you see on midday TV, where they're trying to figure out who the father is. So this is obviously a simplified view of only a few genes, but the mother is donating these peach-colored genes to the child, some of them at least. She doesn't donate all of them. And then one of these fathers is donating the other genes. So here is a band that matches with individual 1 and 2. Here's a band that only matches with individual 1. And here's another band that matches with individual 1 and 3. And so from that, we can determine that the father of this child is individual 1. A second application for DNA technology is medical diagnosis. So the identification of a pathogen or a disease-causing molecule through DNA testing or diagnosis of a human genetic disease. So identifying what kind of disease is infecting you, or if your DNA contains genes for a particular genetic disease that maybe you haven't contracted yet. So applications for this would be viral diagnostic tests. So when you go into the doctor and they do a nasal swab and determine that you do have COVID, or you do have the flu, or you do a COVID test at home, they are often looking for DNA sequences. Some of the COVID tests work differently, but they can identify the DNA sequence of the particular pathogen that's infecting you. Alternatively, it can be used to identify whether you have genetic markers for diseases like Huntington's disease or sickle cell anemia that don't necessarily show very clear symptoms early on. Nasal swabs done for flu and COVID tests require PCR to identify the type of viral DNA collected, because you're only collecting a small sample from the nasal swab. So PCR is used then to amplify that sample or make copies of it so that the tests can be run. And different strains of a virus can also be identified using DNA analysis. So maybe several individuals are coming in with different strains of COVID, and they can be identified and compared. Third, it can be used for forensics and archaeology through DNA testing of human remains or crime scene evidence. So applications here would be if blood, hair, or tissue from a crime scene can be identified or matched to a suspect. So again, you're going to need to be able to compare the DNA left at the crime scene to an actual suspect. You can't just analyze the DNA and figure out who it is. But sometimes DNA is used on ancient human remains and gives clues to ethnicity and ancestry. So it can help to determine whether the remains in an archaeological site might belong to a particular culture or may be a particular monarch that they've been looking for. And just a side note here that red blood cells don't have nuclei with DNA. White blood cells are used for forensic DNA analysis. So those are also found in any type of blood smear, but they're not actually using the red blood cells. Fourth, tracking a disease outbreak. So finding the source of a pathogen or infectious material, or tracking how a disease changes over time. For example, how COVID-19 changed throughout the pattern of infection. Applications here are that infected food can be recalled. So finding the source of a pathogen, if we're looking at a bunch of different samples of lettuce that have been sold in a particular grocery store, if we can analyze the pathogen that's infecting them, analyze the DNA, then we can determine what exactly is causing that infection and how it might be affecting the population. And then that food that's infected can be recalled, knowing exactly what is what has infected the food. So is it bacterial? Is it viral? And then therefore, how can we inform the population about what has happened to their food? Also, infectious diseases can be traced as they mutate. Obviously, we got to see this during the pandemic as the COVID-19 virus was mutating throughout the extent of the pandemic, and the CDC was monitoring the different strains as it mutated through DNA analysis. The flu virus also mutates rapidly, and that's why a flu virus is made every year. It is made to match the most common strain or the strain of the flu that they anticipate will be infecting the population that year. And finally, trade and agriculture. Choosing genetically superior crops, so crops that might have a higher yield or are more pest resistant. We can determine which genes are beneficial by identifying the DNA and analyzing the DNA of crops that are doing particularly well. And tracking and verifying commodities like food. And I'll give you an application of that in just a second. So here are some applications, verifying the labeling of consumer goods. Again, I'll give you an example here in just a sec. And tracking the best genes for certain crops. So here's an example of verifying consumer goods through DNA analysis. In 2013, researchers found about 1/3 of the fish in the United States is not correctly labeled. Merchants often mislabel cheaper fish as more expensive ones to get a better price, Without DNA testing, this may never have been discovered. Consumers often can't tell exactly what type of fish they are eating or purchasing because they look similar or they taste similar, and DNA analysis will help to identify whether there's fraud on the merchant's behalf. So in summary, DNA analysis has many practical applications, such as determining family relationships, diagnosing and tracking diseases, identifying criminals, and verifying consumer goods. In this section, let's discuss how genes can be changed. So we've discussed how DNA can be analyzed. How can it be changed? Well, selective breeding was the first way that genes were modified by humans. So genetic variation always exists in populations, causing individuals of the same species to be slightly different. And humans often encouraged individuals with preferred traits to reproduce through selective breeding. So by breeding individuals with traits that we desire, we end up with more of the population that has those traits. Modern corn is a result of thousands of years of selective breeding. The ancestor plant, teosinte, had tiny kernels encased in a thick covering like this, and eventually through selective breeding over thousands of years, they were bred for larger fruit or corn production, and you can see the scale of the quarter here. So they used to be much, much smaller, and they've been bred for higher yield over thousands of years until we finally have the corn that we produce today. Selective breeding relies on hybridization, the crossing of two different individuals to cultivate the best traits of both. Golden doodles are hybrids, a cross between a golden retriever and a poodle to gain the best traits of both breeds. So they get the attitude of the golden retriever, the reduced shedding of the poodle, and those traits are bred together by humans, and so that's selective breeding. Unfortunately, selective breeding can be very slow. The cassava plant forms a root similar to a potato that is a staple food item for over 500 million people. Although scientists are using selective breeding to encourage the best plants and yields, the cassava only flowers once per year, so selective breeding and hybridization is extremely slow. That flowering process is how they are cross-pollinated for the best traits, and so that is going to be an annual process of hybridizing those plants and breeding them, and that is going to make the cultivation of the traits that we want incredibly slow. So there are faster ways that scientists can change the genetics of a population, and that's through the editing of DNA. New advancements in genetic engineering allow favorable genes to be replicated much faster. So rather than using selective breeding and just pollinating plants with our favorite traits, we're actually changing the genes, which is a much quicker process. When restriction enzymes cut a piece of DNA, sticky ends often remain. So I mentioned that earlier. These are the sticky ends here with a tail of a couple of nucleotides on each side of that cut. This allows geneticists to insert other pieces of DNA, and the combined pieces are called recombinant DNA. So here's a sticky end, and here's another sticky end on the other side of that DNA molecule. And because the restriction enzyme doesn't cut in the same location on both strands, the sticky ends remain where other DNA fragments can be added. So scientists can add a gene that has a complementary strand to this sticky end, and it will actually be brought in and inserted into this DNA. And that is called recombination. Bacteria can pick up small circular pieces of DNA called plasmids in a process known as transformation. So this is just something that bacteria are naturally particularly good at. They incorporate genes from their surroundings, which is one of the reasons that they are able to adapt to new surroundings and cause resistance so quickly, because they are swapping genes and incorporating new genes all the time. So if we can use this to our advantage, if a gene is inserted into a plasmid, then the bacterium will take in that plasmid and copy this gene that we've inserted into it through the sticky ends of recombinant restriction enzymes. And the bacterium will copy that DNA, that inserted DNA, every time it copies that plasmid. The resulting transformed bacteria will produce the protein that the gene codes for, which can then be extracted for human use. So the ability to make human insulin using E. coli bacteria has transformed treatment options for diabetics by inserting that human insulin gene into the plasmid and making the bacteria copy that over and over again. The insulin can then be extracted because the bacteria are making that protein, and then insulin is then able to be transmitted to humans. Another process of biotechnology is called CRISPR-Cas9. So what if there isn't a restriction enzyme that cuts where you want? Because we know that each of those restriction enzymes cuts in a specific location, but that might not be where you actually want to insert the gene. CRISPR-Cas9, a genetic tool only discovered about 20 years ago, can cut at any location on a DNA strand. The CRISPR-Cas9 system is a combination of the Cas9 enzyme, which is responsible for cutting the DNA strand, and an RNA fragment that acts as a guide to locate the target sequence. So locating the set of nucleotides that allows that DNA to be cut in the location that the scientists are looking for. After the DNA is cut at the target sequence, it can be repaired using donor DNA with a different gene, thereby changing the original DNA sequence. So here we're looking at the Cas9 system. We have a target sequence of DNA, and the guide RNA is brought in and it matches at that exact target sequence. This is the Cas9 enzyme here that's going to do the cutting right here at that cleavage location. And some of the DNA is then cut and can be removed, particularly if we're trying to remove harmful genes. And then a different piece of DNA can be inserted in there. So this Cas9 system allows us to cut the DNA at any location, as opposed to using the restriction enzymes, which only cut at a certain location. So this actually allows us to remove particular portions of genes instead of just inserting new ones. Applications for CRISPR technology could include the following and many, many more, because we are truly editing genes with the system. Replacing disease-causing genes with healthy ones, like those for sickle cell, Alzheimer's, muscular dystrophy, Huntington's disease, etc. Any disease that's caused by one gene, that gene could be removed, and then that gene, that particular disease, won't exist in that individual anymore. We could also add in favorable genes to crops for better yield and greater disease resistance. And another example might be removing the allergy-causing gene from peanut butter. So the gene in the peanut that's causing the allergy in humans could be removed, making it hypoallergenic. So in summary, humans have been altering genetics through selective breeding for thousands of years. The use of plasmids and the CRISPR-Cas9 system are new and more efficient ways to transform the genetics of an organism. When genes are transformed, creating a new organism, we call that a transgenic organism. So a transgenic organism is one whose genome has been altered by the addition of DNA from a different species. Foreign DNA can be injected directly into plant and animal cells or added through an infectious agent like bacteria. Sometimes this DNA gets integrated into the host's chromosomes. And sometimes we do this on purpose. In 1990, human genes were injected into bovine embryonic cells. Herman the bull's offspring produced small amounts of lactoferrin in their milk, which is a human protein that can be used to create HIV treatments. So this was an instance or an example of an animal being modified to produce something that was beneficial to humans, which is frequently the way that transgenic organisms are used, but they can also be used in other ways as well. One of the ways that we test the processes of changing genes is through cloning. Cloning is a process in which an exact genetic copy of an organism is made. This can occur at the single gene level through PCR, so PCR is technically cloning, or at an organism level, such as in the case of Dolly the sheep. Now, Dolly was the first mammal ever to be cloned from an adult cell. She was born in 1996 from a surrogate mother using a technique called somatic cell nuclear transfer. So this is the process of cloning at an organism level, and we'll go a little bit deeper into somatic cell nuclear transfer in just a second. In somatic cell nuclear transfer, the nucleus is removed from an egg cell and replaced with the nucleus of an adult body or somatic cell. So this is exactly what this word sounds like, somatic cell nuclear transfer. The nucleus is being transferred and a somatic cell nucleus is being inserted instead. And the cell is then artificially induced to replicate, and the resulting embryo is implanted into a surrogate mother until birth. So let's take a look at this. We have a typical egg cell from any female organism with a nucleus. If we eliminate the nucleus of the egg cell, it's basically a blank egg cell with organelles but no genes. If a somatic or body cell nucleus is inserted into that egg cell, the difference here is then that the egg cell started, let's say we're discussing humans, it started with a haploid number of 23 chromosomes, and the somatic cell nucleus has 46 chromosomes. So this is now a full set of chromosomes. It does not need to join with any other cell. It has all of the formatting of an egg cell, but it has the genes of an adult cell that has already joined with a different organism in sexual reproduction. And through artificial stimulation, we can make that egg cell divide into an embryo. And that embryo is then usually implanted into a surrogate mother until that cloned organism is born. And that cloned organism will be a copy of whatever this somatic body cell was removed from. So whatever organism this nucleus came from, that is the clone that's going to be created, because those are the genes that are in that egg cell. So the resulting baby is a clone of the adult donor organism. So here's an example. If an egg cell from a black-faced sheep is joined with an adult somatic cell from a white-faced sheep, it's then implanted into a black-faced surrogate mother. What color is the baby cloned sheep going to be, and why? Think about this for a second. So the cloned organism is going to be white-faced because the adult somatic cell donor that donates all of the genes for that egg has a white face. So that individual clone baby is going to have that white face as well. The egg cell donor and the surrogate mother do not donate any genes to this cloned baby, and so they are not going to have an effect on that baby's phenotype. So that's all well and good. It's fun that we can clone things, but what is actually the purpose of cloning? So although it seems like science fiction experimentation, it does have some valuable applications. First, livestock and crop cloning can reproduce favorable individuals more quickly and without the mixing of genes that occurs during sexual reproduction. So this is like artificial asexual reproduction, not mixing of genes, not contaminating them potentially with genes from another individual through sexual reproduction. Pets could be cloned after death. This is a possibility that a lot of individuals are interested in. Infertile couples could create a clone of one parent rather than joining the genes of both parents. They could create a clone of simply one. And tissues and organs can be cloned to use in the event of a degenerative disease. So this is one of the applications that has the highest potential for creating cloned tissues and organs from healthy individuals that can then be transplanted into diseased individuals or individuals that are having a breakdown of some of their body systems. So in summary, transgenic organisms incorporate DNA from more than one species. Cloning creates exact copies of a gene or an organism, and somatic cell nuclear transfer is one process of creating these clones.